Hello and welcome back. We're now at a critical stage of the CRISPR process where the DNA strand has been cut at the target site. This cut, made by the Cas9 protein, creates what's known as a double-strand break, or DSB. So, what happens next? Well, the DSB serves as a trigger for the cell's natural DNA repair mechanisms. These mechanisms are essential for maintaining the integrity of the genome, but in the case of CRISPR, they're exploited in order to introduce specific genetic changes. Let me walk you through the two major pathways cells use in order to repair the DSBs. First is the non-homologous end-joining pathway, and second is the homology-directed repair. First, let's talk about the NHEJ, also known as the non-homologous end joining. This is a quick and very efficient repair process wherein the cell simply joins the broken DNA ends together without using a repair template. And here's how this works. A specialized repair protein recognizes the broken ends of the DNA. They bind to these ends, process them, and they link them back together. Sounds straightforward, right? But here's the catch. During the repair process, researchers purposely perform small errors or alterations. Now, these alterations either take the form of A, insertions, where extra nucleotides are added during the repair, or B, deletions, where certain nucleotides are removed. Collectively, these changes are called indels, short for insertions and deletions. They alter the original sequence of the DNA, and this has multiple important impacts. Let's now look at the different impact of these indels. First is frame shift mutation. Now, if the indels occur within a gene's coding region, they can shift the DNA's reading frame. This essentially changes how the gene is read, and this oftentimes leads to the creation of non-functional proteins. Second is through premature stop codons. Now, indels can perhaps introduce stop codons, which are essentially signals that tell the cell to stop building a protein prematurely. Now, this essentially truncates the protein, making it very incomplete and non-functional. Third is through gene slicing. Now, when the NHEJ introduces significant disruptions, the gene may stop functioning entirely, and this is referred to as a gene knockout. Now, at this point, you might wonder, why would researchers deliberately create these disruptions? Well, these alterations help us develop a greater understanding of DNA and of gene function. Let me explain this with an example. Now, through gene knockout, researchers can essentially A, deliberately disrupt a gene in order to understand its role and its biological processes, but B, eliminate the harmful effects of a gene, such as those carried by perhaps disease-causing genes. So NHEJ is incredibly useful for studying gene functions and for applications like disease modeling. Now let's move on to the second major DNA repair pathway, homology-directed repair, or HDR. Now this process is way more precise than the NHEJ and is widely used when scientists want to make specific edits to the genome. Unlike the NHEJ, HDR relies on repair templates in order to guide the repair process, and here's how it works step by step. A. In terms of the repair template, scientists introduce a synthetic repair template into the cell. Now, this template contains homologous regions. These are essentially DNA sequences that match the DNA flanking the DSB, and they trick the cell into using the repair template during the repair process. Second part is desired modifications, where between these homologous regions, the template includes the genetic changes that the researchers purposely want to introduce. Now, these could be corrections to a mutation, perhaps insertions of new sequences, or even subtle changes to the genome. Now, the second is the repair process, wherein the cell uses a homologous region to align the repair template with the broken DNA. It then incorporates the desired modification into the genome as it repairs the DSB. Now that brings us to the end of this video. Thank you for watching, and I hope you developed a better understanding of the CRISPR technology.